Shri Gurubhyo Namaha children. I am happy to welcome you all for our online teaching academy Ekalaiva. For the class 12, subject Biobotany, chapter 4, Principles and Process of Biotechnology. Today we are going to discuss about vectors. Another major component of gene cloning or rDNA technology, vectors are very essential. They are transporting material. Okay, so vectors such as plasmid is a very important component in rDNA technology. A vector is a small DNA molecule. It is also a small DNA molecule capable of self-replicating. What is meaning of replication? The same carbon copy, the DNA replicate to form another DNA, that is called replication. So vector is also having the replication property. So vector is a small DNA molecule having self-replication and it is used as a carrier. So all the vectors are carriers. They carry some gene of interest from one organism to another organism. You get my point, children? Yes. So vector is a small DNA molecule capable of self-replicating and it is used as a carrier and transporter of DNA fragments which is inserted into it for cloning experiment, for gene cloning experiment, for rDNA technology. All the things the same meaning only. Clear? So vectors is also called as cloning vehicle or cloning DNA. Why it is said to be cloning DNA? Vector is nothing but a small DNA molecule only, no? So it is said to be cloning DNA. It is involved in gene cloning, so it is said to be cloning DNA. Are you get my point, children? Yes. Vectors are of two types, children. One is cloning vector, another one is expression vectors. So what is cloning vectors? Cloning vectors is nothing but it is a vector used for cloning of DNA. Okay, so cloning vector is used for cloning of DNA insert inside the suitable host cell. And another type, expression vector. Expression vector is used to express the DNA insert for producing specific protein inside the host cell. What is the connection between DNA and protein, children? If there is any link between DNA and protein? Yes, they are having some relation, the link between DNA and protein. Have you heard the word central dogma? What is central dogma? Here it is DNA. DNA have the property of replication. They produce the same DNA, another DNA, daughter DNA, and then DNA to mRNA. So conversion of DNA to mRNA, it is called transcription. Okay, now conversion of this mRNA to protein formation, that process is called translation. Clear, children? Now DNA have the link to form protein, that is called central dogma. Conversion of DNA to DNA, that is called replication. DNA to mRNA, that is called transcription, and mRNA to protein, that is called translation. Okay, so expression vector is used to express the DNA insert for producing specific protein inside the host cell. Clear? Now we will see the properties of vectors. Vectors are able to replicate autonomously. As I said, a vector is a small DNA a small DNA segment. So once it is said to be DNA means they have a very important property of DNA, self-replication. So vectors are also having the ability. So vectors are able to replicate autonomously to produce multiple copies of them along with their DNA insert. If any DNA is inserted inside the vector replicates itself along with the DNA insert. Clear, children? The next one, it should be small in size. Usually vectors are small in size and of low molecular weight, less than 10 kilo base pair in size, so that it is very easy to enter or easy to transfer into the host cell. Okay, so the size may, if it is very big size means it feel very difficult. We feel very difficult to insert the vector into the host cell or host plant. Clear? Yes. Vector must contain an origin of replication. There will be a site called ORIC, that is origin of replication, so that it can independently replicate within the host cell. Clear? So vector should have unique target sites for integration with the DNA insert. Then most of the commonly used vectors, cloning vectors, have more than one restriction sites. So these are the very important properties of vectors. Then types of vector, children. First one, very important vector, we often using this vector. The name of the vector is plasmid. Very important question. Okay, plasmid. Plasmids are extra chromosomal, self-replicating double stranded DNA. As I said earlier, vector is a DNA segment. So types of vector, it is also, plasmid is also a vector. It has all the properties of the same vector. So plasmid are extra chromosomal, self-replicating double stranded circular DNA molecule found in the bacterial cells. So the plasmids is a type of vector. It is a double stranded circular DNA self-replicating vector having extra chromosome which is present in the bacterial cell. Clear? So once again I am repeating the same. Plasmids are extra chromosomal, self-replicating double stranded circular DNA molecule which is found inside the bacterial cell. See here, children, this is a bacterial cell. Bacteria contains the bacterial DNA. Okay, along with that a double stranded circular self-replicating DNA, that is called, very famous one, plasmid. Clear? And then plasmid contains genetic information, all the genetic information for their own replication. So genetic information is nothing but DNA. Okay, so plasmid contains all the genetic informations for their own replication. Clear? See the picture of bacterial cell. This is a bacterial DNA and this is a plasmid. Clear? Then first types of plasmid, very example for plasmid, that is PBr322 plasmid. This is one type of plasmid. Okay, example for plasmid. PBr322 plasmid. The name itself you feel somewhat complicated, right? Why that much of complication here? Nothing. P, small p, BR322. There will be so much of story in that name, PBR322. Let's see. PBR322 plasmid is a reconstructed plasmid and most widely we often using this. Okay, so reconstructed plasmid and most widely used as a cloning vector. It contains 4361 base pairs. Just imagine base pairs, 4361 base pairs. How the name comes PBR? What's special in that PBR322? Just think. See here. In PBR322, PBR, the P, the letter P denotes the plasmid and the BR B and R respectively the names of the scientist. Please listen here. The scientist name Bolivar and Rodriguez. They are the two scientists discovered the plasmid PBr322. So it is named as P represents, small p represents plasmid, capital B and capital R represents the scientist name, the first letter of the scientist name, Bolivar and Rodriguez, who developed this plasmid. They only developed this plasmid, so they named as PBr. Then what is 322? Let's think. The 322, the number 322 is the number of plasmid developed from their laboratory. They developed 322 plasmids in their laboratory, so they named as plasmid PBr322. See the picture here, children. It contains AMPR. What is AMPR? Ampicillin resistant gene, and then TTR, TETR, tetracycline resistant gene. So it contains ampicillin resistant gene and tetracycline R resistant, two different antibiotic resistant genes, and also so many restriction sites, sites for several restriction enzymes. Example, HIND3, ECO, R1, BAM, H1. These are all the examples of restriction enzymes. So many restriction sites are also there in the plasmid vector. See the picture here, children. See, this is AMPR, ampicillin resistance gene, and tetracycline resistance gene is there. Apart from that, several restriction sites for binding of the restriction enzymes. What are they? See here, ECO, R1. These are all the enzyme restriction enzyme. So many restriction sites of several restriction enzymes. So ECO R1, then PVU1, PST1 and BAM H1. These are all the examples of restriction enzymes. So they are the sites for restriction enzymes. Clear, children? The second vector is the types of plasmid is TI plasmid. It is also very important. Okay, so TI plasmid is the name of plasmid, is found in Agrobacterium tumefaciens. Agrobacterium is nothing but the bacteria which is present in the soil. It is also called a soil bacterium. Clear? So Agrobacterium tumefaciens, soil bacteria. It provides strength to the soil. Okay, so this plasmid is extracted, taken from this particular bacteria. Okay, TI plasmid is found in Agrobacterium tumefaciens, a bacteria responsible for inducing tumors in several dicot plants. The plasmid carries transfer gene. This plasmid always carries transfer gene, which helps to transfer tDNA of one bacterium to another bacterial or other bacterial cell. Clear? So see the picture here, TI plasmid. It is also called as tumor inducing plasmid in plants, TI. So TI plasmid is found, it is present in the Agrobacterium. Agrobacteria is nothing but a type of bacteria present in the soil. Clear? It has ONC gene, ONC, ONC gene for oncogenicity, cancer inducing gene. Okay, oncogenicity, and ORI gene, ORI gene. ORI gene means that is site of replication, origin of replication. So ORI gene for origin of replication and INC gene for incompatibility. tDNA of TI plasmid is stably integrated with plant DNA. Agrobacterium plasmid have been used for introduction of genes of desirable character into the plant. So we just suppose had some any character, important or desirable character with this plasmid, and it is inserted into the plant. Okay, so Agrobacterium plasmids have been used for introduction of genes, of desirable characters. Okay, see the picture here. The next one is transposon or as vector. Transposon is also a DNA sequence or DNA sequence able to insert itself at a new location in the genome without having any sequence relationship with the target locus. Here transposon are also called as walking gene. See here, very important 2 more question. What is walking gene? Why it is called so? Clear? So it is also called as walking gene or jumping genes. They are used as genetic tools for analysis of gene and protein functions that produce new phenotype on host cell. The use of transposon is well studied in Arabidopsis and also in bacteria such as E. coli. So here this Arabidopsis and E. coli, this particular transposon is used in many ways. So the use of transposon is well studied here in E. coli bacteria. See the picture of a transposon. This particular part is said to be transposon. It is a segment, DNA segment. The next one is expression vectors. Vectors which are suitable for expressing foreign proteins is said to be or is called as expression vectors. This vector consists of signals that is necessary for transcription as well as translation of proteins in the host. I already said what is transcription. Transcription is nothing but conversion of double stranded DNA into single stranded mRNA. That is called transcription. Clear, children? And translation of protein is nothing but conversion of mRNA to protein. That is called translation. So this particular vector, what type of vector it is? This is an expression vector. This vector consists of signals. So many signals are there in the vector. It is necessary for transcription as well as translation of proteins in the host cell. Clear, children? Yes. Next, this helps in the host. This vector helps in the host to produce foreign protein in large amount. So example for expression vector is PUC19. Clear? So see the picture here. This is the picture of expression vector. Here so many the sites is there. A restriction site is there, and then here see the transcription initiation sequence is there, and then expression vector consists of certain operon sequence and particular DNA sequence is there. Here the site of transcription starts, termination of transcription site is here, and then origin of replication is here. So expression vector is nothing but it is a type of vector suitable for expressing foreign proteins. That is a main key points, very essential for expressing the foreign proteins. Okay. Thank you, children. We will see in the next class.